Today we are starting a series of lectures on microbiology. So let's start with the very basic concepts, right? For example, what are the major categories of infectious agents, right? We'll discuss about the different categories of infectious agents. Start with the smallest one. Yeah, which is the smallest infectious particle? Question goes to someone very bright, Dr. Miguel.
Prions.
Prions. What are prions? Prions. So Mr. Miguel is telling us the smallest infectious particle is prion, right? So Dr. Sunny is going to tell us that prions are having DNA or RNA. Yes, please. They are having DNA. Dr. Sunny claims that prions have DNA. Anyone who agrees to Dr. Sunny? Only Sunny is agreeing to himself. Okay. People who believe prions have RNA, raise their hands. Okay, now listen. People who believe prions have DNA, Sunny is alone. And people who believe that prions have RNA, raise their hands. Okay, Jose Bird, Jose Rani. You must remember, Sunny is wrong. And you must also remember, Jose Bird and Jose Rani are wrong. These are infectious particles which do not have any DNA or RNA. That's a very important thing to remember. They are just protein particles. They have no recognizable molecules of DNA or RNA. I think it must be shocking for you, isn't it? But this is true, that there are infectious particles which do not have any DNA or RNA, but when prions enter your body, for example, if they reach your central nervous system, few prion molecules or few prion particles, eventually there will be billions of them there. Question is, if prions don't have any DNA or they do not have any RNA, how they increase their number? Yes, Dr. Ammon is going to tell us. Prions don't have any DNA, they do not have any RNA, but once prions enter, suppose, in your central nervous system, over the time they convert into billions and billions of their copies. How their number increases? Yes, Miguel.
They use the host's DNA and RNA to replicate themselves.
Okay, that's right. He's saying that prions enter into our neurons, suppose, and you think that prions will use our genes to make more prions. And no, prions do something like that, but not exactly like that. Let me tell you exactly what happens. Very briefly about prions. Basically, prions-related proteins are normal proteins present in our body. Let's suppose here is a neuron. Because prions are producing a group of diseases which are called, I will explain how the prions number increases, right, when they do not have their DNA or RNA. But before that I must tell what type of diseases they are producing. The group of diseases which are produced by the prions, they are called, yes, transmissible, yes, spongiform, encephalopathies, right? So how the prions really increase their copy? Let me tell you. Actually, prion related proteins are normally produced by our cells. Prion related proteins are normally produced by our cells. They are called prion protein from cellular region, PRPC, prion proteins from our cells. Let me show you. This is your DNA, right? And within your DNA, you are having a gene, and when this gene expresses, it will make messenger RNA, and that messenger RNA, when it is translated, it will produce, I'm talking about normal gene first. This normal gene, when it will produce messenger RNA, it will produce a protein, and let's suppose this is that protein, right? Now this protein which is produced, of course it is amino acid sequence in a string, and they are arranged in a spring or in a coil fashion, or such arrangement of peptide chain is called helical arrangement, alpha helical arrangement. Is that right? So normally our cells have genes which can produce prion related proteins. These cellular prion related proteins, right, these are arranged in alpha helical arrangement. They are arranged like their peptide chain is in a spring shape. Am I clear? Now look at it. What are prions which are pathologically abnormal? They are the same protein but they are misfolded. What are prions? These are infectious particles which are the same normal protein but abnormally folded. Is that right? As I told you that normally this protein should be folded in spring. I will tell you how exactly normally it is folded. This is the arrangement of protein normally. But if you misfold this protein, the same peptide chain is misfolded, and rather than this coiled arrangement, rather than this alpha helical arrangement, if it is, okay, I will make with the same color so that you must know. Now, this is a peptide chain, that is also a peptide chain. Amino acid sequence in both chain is absolutely same. It means the primary structure of the protein is absolutely same. But this is folded in a coiled fashion and this is folded in a different way, and this arrangement is called beta plating. What is this arrangement called? Beta plated arrangement. Beta plated arrangement. So what really happens? When normal helical proteins, prions, right, they are beta plated, they become abnormal proteins. Now question is that why this is abnormal? Why this is able to produce disease? Is that right? Answer is that normally in our cells many proteins are being produced and catabolized. You know, there's a protein turnover going on. When genes are producing the proteins, enzymes, or different structural and functional proteins, with the time when those proteins become denatured, those proteins are destroyed and they're replaced by more proteins. Is that right? This is a normal protein turnover. Now, it means for normal protein turnover, cells should be able to produce the protein as well as should be able to destroy the protein. And to destroy these proteins, there must be proteolytic enzymes which should be called proteases. What should be called the? Proteases as a group. So every cell has proteases which will destroy eventually all denatured proteins. Am I clear? Now, all the proteases which are present in human cells, they work on mainly on the alpha helical arrangement. They do not work effectively on beta plating arrangement. That is the real concept, that the enzymes which are present in our cells or in our tissues which can break down the proteins, they break down alpha helical arrangements, they cannot break down beta plating. Is that right? Now, if due to any reason in your cell or outside your cell, lot of peptide chains are present in beta plating, can you catabolize those proteins? You cannot. Should I give you classical examples? You know silk suture, do they absorb? No, why they don't absorb? Silk is a protein. Yes, Miguel. You never thought of it. Oh my God. Silk is produced by the silk worm, you know. Silk is having beta plating, that is so simple. Silk, S-I-L-K, right, has having beta plating arrangement. That is why when you put suture with silk, our enzymes cannot digest it away, they become permanent. So look at it. What are prions? They are something like silk. Just to have a concept in your mind. These are misfolded proteins. These are corrupted proteins. You know, as from some society it's very difficult to root out the corruption, in the same way it is very difficult or almost impossible to root out the prions out of the cells because of beta plating. Is that right? No problem up to this? Do you know any other protein which is beta plating? You must have heard of diseases other than prions. Have you heard of the group of diseases called amyloidosis? Amyloidosis, amyloids. What are amyloids? Have you heard of amyloid? Of course, what is amyloid? Any protein in your body, yes, which is beta plated and present extracellularly. That protein is called amyloid. You are understanding? That is why once amyloids start depositing in your tissues, you cannot digest it away because they are resistant to proteolytic activity. Am I clear? Let's go back to prions now, having this basic concept. Look, what are prions? Now, onward, I will not make whole this structure. I will just show alpha helical arrangement in this way, and I will show the beta arrangement in this way, now onwards. Now, what are prions? Prions are basically infectious particle, infectious agent. What really happened? They are beta plated misfolded protein particles. You must have heard of something called mad cow disease. What was happening there in that disease? Right? In mad cow disease, which is also called bovine spongiform encephalopathies. Mad cow disease, bovine spongiform encephalopathy. The infectious agent of mad cow disease is prions. What was happening that if from the cow meat, these prions enter into your body and these prions reach to your central nervous system, what will happen? Look here. This is the mad cow disease agent has entered into this neuron, right? This is pathological particle. This is corrupted protein. Now, what this will do? Normally, these genes are expressing and producing normal prion-related cellular proteins which are folded in a normal fashion. Now, a corrupt protein comes. This protein will actually alter the folding of the normal proteins. How? That when, look, when messenger RNA come, primary, then on the ribosome, primary peptide chain is made, then you know, primary peptide chain enter into endoplasmic reticulum and into Golgi operators, and there is modifications going on into endoplasmic reticulum and Golgi operators. And during that, proteins are appropriately folded, and those changes are called post-translational modifications of peptide chain. What are they called? Post-translational modification of peptide chains, because first peptide chain is translated, messenger RNA is translated into peptide chain. After that it undergoes folding. Now, in the presence of beta plated particle, now this is a very important concept, listen with your both ears. In the presence of abnormal pathological prion particles or in the presence of beta plated prion particle, the newly formed proteins are misfolded. As you know, in one department, as it happens in government department, a big corrupt man come and slowly all the department become corrupt. What happens? Even the people who are not corrupt, gradually they start becoming corrupt. In the same way, this is a corrupt protein which enters and it influences our own normally produced proteins and corrupts them into misfolding and convert them into prions. So what really happens, this protein and that protein, they come together, right? They come together and in the end, you will end up with two prions. Then these two prions will influence two more normal proteins and then you will end up with, yes, 4 prions. He knows mathematics. Good. So, 4 prions. So now you understand that we had originally 1 prion enter into this cell, but it started corrupting our own proteins and converting them into pathological beta-plated particles, and when a lot of beta-plated particles will accumulate, cells cannot digest them and you can say cell become a garbage house, right? Because the number 1, beta-plated, these proteins are not functional, and number 2, which cell is unable to digest them, and because normal prion proteins are protease sensitive. Pathological misfolded beta-plated prion proteins are protease resistant. So what is the real difference? Protease resistance particles which are pathological, protease sensitive particles which are physiological. So this pathological particle convert our physiological particles, normal particles, into corrupted proteins. This is what happens into prion. The whole purpose of this discussion was that prions are the smallest infectious agent known up to now, and they don't have any DNA or any RNA, but still while entering into your body, they can increase their number into billions and billions, right? Not by giving their own offsprings, rather they just start corrupting your own prion related proteins, right? This was a little introduction to prions. We'll discuss into detail later on about prions. Okay. So, and I always remember, it's worth remembering and worth repeating that beta-plated proteins which are heavily beta-plated, our body cannot digest them away, and pathological conditions related with beta-plated protein, one is prion related diseases, other is amyloidosis, right? Now, we come to the next group of infectious agents, right? Who is going to tell me? Yes, please. Virus. I think something before virus. Okay, I will put virus so that you feel somewhat comfortable here. This is virus. Okay? Something less than a virus can also produce disease in an indirect way. Or they are responsible. Now you are shocked. Why you are looking shocked? We are talking about infectious diseases. Yeah. You are, I think, in romantic love with virus and bacteria. There are other things also which produce infections. Anyone? No one want to speak anything? Some agents which are infectious, or directly or indirectly they can produce some infectious problems for us, or produce some virulence, or produce some disease. Why don't you? Oh, he is telling toxins. My friend, toxins are not infectious agent. Right, we'll talk about those later. Let me tell you, have you heard of something called bacteriophages? Something called plasmids? Something called transposons? I will explain that, right? How they are infectious agent. bacteriophage bacteriophages, plasmids, and transposons. Let me explain what I really mean by this. Let's suppose take an example of plasmid. What is plasmid? What is plasmid? You never heard of it? I'm about to die with shock myself. What is plasmid? My God, it's a dangerous situation. Plasmids are, usually they are present in bacteria. Let me show you. Here is a bacteria and I will explain these things in relation to bacteria. Let's suppose here is bacteria and this is bacterial DNA. Right? Plasmid is, this is bacterial chromosome. Plasmid is extra chromosomal DNA, double-stranded circular DNA. Extra chromosomal, this is very important. Plasmids are extra chromosomal double-stranded DNA ring, which are capable of replicating on their own. These are double-stranded DNA rings, circles, right? And if one plasmid suppose enter into this bacteria, after some time, there may be maybe 100 plasmids here. And then this is another bacteria. Here is another bacteria. Let me explain how plasmids affect the disease process. Bacteria number 1, bacteria number 2, and bacteria number 3, right? Which is the most abundant organism in the world? Viagne. Don't tell me human beings. Which is the most abundant organism in the world? Miguel. Anyone? Bacteria. You put all the animals and all the plants together and make a big lump. Bacteria are more than that combined lump on the earth. Right? Anyway, let's come back. So what is happening? This is bacterial chromosome. This is extra chromosomal double-stranded DNA which has the capacity to replicate on its own. For example, this chromosome may not replicate, but plasmid may keep on replicating independently and make extra copies. Am I clear to you? You understand what is plasmid? Now, what is the clinical relevance of plasmid? Look here. This is, I'm just giving you basic idea. Later on, we'll have a full lecture on plasmid. Right? This is just an introduction to microbiology. Now what happens? Let us suppose in this plasmid, this is a gene. In this plasmid, let us suppose this is a gene, and when this gene expresses, a special toxic molecule is produced, a special toxin is produced. It means if this bacteria is not having that plasmid, can this bacteria produce that toxin? If this bacteria cannot produce that toxin, let us suppose this bacteria is not virulent. So now, this is a virulent bacteria because this bacteria is a special type of plasmid, and plasmid is having a genes to express special type of toxin molecules, right? So this is a virulent bacteria or bacteria capable of producing disease. This is another bacterium which is not having that plasmid and it is not having any other virulence factor, so it is not able to produce a disease. Now you imagine, if the plasmids from there transfer here to this bacterium, right? Later on we'll talk how plasmids transfer. For a while you just trust me, they do transfer. If from this bacterium that this plasmid shift to another bacterium and now plasmid come over here, so what will happen? Yes please. That now this bacterium will start producing, yes please, toxins, and it will convert into which type of? Virulent, and they will produce the disease. And now this bacteria will be able to produce infection and pathogenicity. So what happened? Maybe this bacteria was part of our normal flora. It was innocent bacteria in us, not producing any disease, but as soon as it got this special type of plasmid, this bacteria was able to express toxins and produce the disease, so this become highly infectious situation. So just plasmid can act as an infectious agent, but through bacteria. Is that right? So plasmid can alter the virulence of bacteria. For example, some other plasmids have other genes, and other genes present in plasmid may, for example, plasmid may have another gene, and this plasmid gene, when this expresses, then a new protein will be made, and that protein help the bacteria to stick to our mucosal cells. And these proteins are called adhesins. What are these? Adhesins. If this plasmid is not there, bacteria don't become adherent, don't produce disease. When this plasmid is there, then they are capable of producing the disease. So it means plasmids are related with pathogenicity. Am I clear? Either by producing toxins or by producing adhesin molecules. Am I clear? Another thing, a real dangerous thing, every doctor must know. Plasmid relationship with the drugs. I'm not talking about illicit drugs. I'm talking about antibiotics. Yes. What is the relationship of plasmid with the drugs? Dr. Jose? No idea. Okay, Mr. Vyagne want to tell us something. Okay, very good. He say that plasmids may produce resistance against a drug. Yes, he's right. Sometimes what happens, let us suppose, this is special type of, just tell me what is this antibiotic I've made? Yeah, what is this antibiotic structure? Please tell me this is penicillin. Oh my God. This is penicillinoic acid ring, right? And what is this ring? Beta-lactam ring. Have you heard of things like this? Right? So what happened? Actually, we use the penicillin to kill the bacteria, but destroy the bacterial cell walls. You must be knowing this thing. Is that right? Now, sometimes what happens that plasmids, some plasmids may have a special gene, and when this gene expresses itself, it makes messenger RNA, and that messenger RNA translates into a protein, and that protein, look at it, this is that protein. What that protein can do? What is happening? This protein produced by the plasmid can destroy the beta-lactam ring. And when beta-lactam ring is destroyed, do you think penicillins can work? No. What should be the name of this enzyme? Penicillinase or beta-lactamase? Penicillinase or beta-lactamase. So some of the plasmids, attention, carry the genes to produce the enzymes which can destroy our antibiotic. So bacteria is happily producing disease, and whenever antibiotic come near, bacteria produces enzymes and destroy the antibiotic structure. And in this way, bacteria become resistant to drugs. So plasmids can also induce resistance in the bacteria against the drugs. Right? So we can say that what are plasmids? Plasmids are extra chromosomal, double-stranded DNA rings which are capable of multiplying independent of the chromosome. Even though plasmids have one more capability, we'll discuss later, they are so nasty, sometimes they open their ring and become the part of our, integrate into our DNA. Right? Anyway, we'll talk about that later. Right now, plasmids are very important concepts of plasmid as infectious agent, but they don't produce infection directly. Just having plasmid in your body will not produce trouble, but plasmids may enter into your bacteria and increase the virulence of the bacteria and indirectly produce the disease. Am I clear? Any question you have? No. Bacteriophages. What are bacteriophages? Yes, doctor. And how they are related with the disease or infectious disease?
Okay, the formula definition is the organism that destroy or it's the bacteria.
Oh my God, she told me that the bacteriophage is an organism.
Or
It is not an organism. It means after a few minutes I will talk about virus is organism or not?
no,
No.
because they are not.
Organisms usually have cells. Right? Bacteria is an organism, of course.
Yes.
Okay, let's come back. Let me tell you how the bacteriophages have a role as in the infectious diseases. Have you heard of a bacteria called Corynebacterium diphtheria? This bacteria is sometimes normally present in some healthy people throat. Now, Corynebacterium diphtheria, right, diphtheria bacillus can be present in two types. Look here. This is diphtheria bacteria, Corynebacterium diphtheria, but it is not producing any toxin, right? Because it does not produce any exotoxin, it is not capable of producing the disease diphtheria. Is that right? Now what happens? Bacteriophages are special type of viruses. I'm going to make a bacteriophage here. This is a bacteriophage. What bacteriophage is doing? Bacteriophage has special type of DNA in it. And whenever it get a chance, it will stick to some bacteria, make a hole and inject its DNA to the, where? Okay, I'll make it different color so you don't confuse it with plasmid. Bacteriophages are special type of virus which are having genetic material, and these viruses interact with bacteria. And how do they interact? Bacteriophages can inject their genetic material into bacteria. Now this is bacteriophage genetic material. Bacteriophage genetic material may have special type of genes. What was this bacteria? Corynebacterium diphtheria. Is that right? And this diphtheria bacillus without this genetic material was not pathogenic. This was not dangerous bacteria. But as soon as bacteriophage is a virus which is capable of injecting its genetic material into a bacteria. This is a very special type of bacteriophage which has attacked this bacterium and injected its genetic material, and genetic material of this bacteriophage had a special gene. This gene is called tox gene. Tox gene. And when this gene expresses itself, a special protein is produced, and that proteins are being secreted by this bacteria, and these proteins are called diphtheria toxins. Diphtheria exotoxins. And these are responsible to produce the damage in the tissue. And these exotoxins produce what? Disease called diphtheria. Now, by this thing you can understand, if this bacteriophage is not there, if this bacteriophage and its genetic material is not there, can this bacterium can produce disease? No. Is that right? Because bacterial chromosome does not have the gene and capability to produce the diphtheria toxin. So every Corynebacterium diphtheria does not produce disease. Only those diphtheria rods are toxic which have this type of genetic material from bacteriophage. Am I clear? So it means bacteriophage have also something to do with the infectious diseases, even though indirectly. Am I clear? No problem? Now, so you understand, bacteriophages can bring the genetic material. Plasmids are themselves genetic material. Of course, this is extra chromosomal. Transposons. What are transposons? I'm about to be impressed by someone. What are transposons? Transposons are the sons of transporters. You know, transporters are transporting the things. Their sons should be very mobile. Transposons are special type of DNA fragments which can jump around. Let me tell you how. Even within a bacterium, let's suppose this is a bacterium, right? This is bacterial DNA, right? Here is your friend plasmid, right? Transposons are special type of double-stranded DNA. This is that genetic sequence which is called transposons. I should show that they are having the legs also because they keep on jumping around. Actually, these DNA fragments have a very special capacity. They can leave from this point and go within the same chromosome at other point, or they may leave from this and go to the plasmid, and from the plasmid they may go to another chromosome. So these are also called jumping genes. What are they called? Jumping genes, transposons, the sons of transporters. They're very, very mobile. We can also call them mobile genes, right? So these mobile genes have to do with the disease also, something with the infection. If transposon is having some genes which are related with virulence factor, for example, transposons are having a gene which can produce toxins or some adhesins or some other protein which can related with the virulence factor, and if transposons jump from one bacterium, for example, transposon jump from this chromosome, bacterial chromosome to plasmid, and then then plasmid goes to another bacterium. So the new bacterium will acquire new characteristics due to arrival of this transposons. You are understanding? They are using the plasmid as a transport system, right? And in this way they can spread within the population of bacteria and alter the pathogenicity of bacteria. Am I clear?